my title is Regulatory Perspectives and How Composition Data Are to be Interpreted Feed. All right? So while we'll talk about other things, the emphasis of my talk is on feed. And just to clear the air, Codex does not cover feed, all right? But it's intertwined with feed, so it's very difficult to separate. So essentially, whatever Codex has published applies to feed, although the Codex Alimentarius publication will say it doesn't apply to feed. And the reason for that, because there's a human and there's an animal part. And the animal part would be what would apply to feed, and there's been no effort in that to work on GM feeds. I thought it was good to talk about terms, which we've been talking during the workshop. Genetic engineering, a process of transforming recombinant DNA technology. Transformation event. Transformation event means the introduction of a plant genetic material that has been manipulated in vitro. And that comes from the FDA 92 policy. Genetically modified or bioengineered means food derived from a plant that is developed using a transformation event. Conventional counterpart, a related variety, its composition or products for which there is experience of establishing safety based on common use as a food. That comes from the OECD documents. Experimental comparator, conventional counterpart. Okay, regulatory mandates of all countries is to ensure the safety of all domestic and imported foods intended for human or animal consumption. Composition analysis of key components is a part of safety assessments. Comes from Codex and OECD documents. Food and feed regulatory process for all GM crops. There are two types of regulatory procedures. One is specific product-based, and that's the U.S. and Canada, where we're most interested in what's being produced that's in the new plant variety. And then there's the specific process-based, and that's the EU and I think Australia, according to publications, where they're more interested in the process as well as the product. Both procedures require composition data. Intended versus unintended changes. Intended changes in new plant proteins, increase or decrease levels of plant components. Pretty straightforward. We've talked about that during the week or during the workshop. Unintended changes, increased or decreased levels of nutrients, anti-nutrients, and/or natural toxicants, and there's some thing called other compounds that are thrown in that come up in the OECD documents. Composition is common to all regulatory safety assessments. For intended effects, the new plant proteins or new or altered metabolites or compounds. For unintended effects, we got nutrients, anti-nutrients, and toxicants, and then the other, which I didn't have listed here. Okay, for the purpose of this talk, I'm not going to talk about the new plant proteins nor the altered metabolites. I think these are all on a case-by-case basis and dependent on what the protein is, the molecular process and all. This is common, and I think for the purpose of this talk, we're restricting it to the unintended effects, all right? In that respect, as Kathleen Jones had told us, OECD has published crop-specific consensus documents that list the key nutrients, anti-nutrients, secondary metabolites, and natural toxicants. For other crops, data on these compounds are extracted from the literature because there are other varieties, plant varieties, other than those that are common, those that are covered by OECD, those that are covered by ILSI database, that we have to find composition data. And those generally come from the literature for that data. And one of the problems is going to be the methods, and Hillary talked about validating methods yesterday, but that has to come from literature with some eye on how good the data is. The U.S. National Research Council has published a scientific report on nutrient requirements of domestic animals, which serves as a standard for deriving key nutrients, anti-nutrients, and natural toxicants in animal feeds. So in the U.S., and I think how many people are familiar with the NRC nutrient requirement publication? Those are on their website. You can go to the NRC website and you can get those. And so we use those in determining what are really important. Key nutrients as defined by OECD. We have proximates, amino acids, fatty acids, calcium, phosphorus. Key anti-nutrients as defined by OECD is adversely affect metabolism. Example, trypsin inhibitors in soybeans. They have those that bind minerals and make them unavailable to the feed, phytic acid in corn. Cause adverse effects to the animals, phytoestrogens in soybeans. Cause adverse health effects in animals, gossypol in cottonseed, cyanogenic acid, or cyanogenic glycosides in cassava. Hydrogen cyanide, makes it simple. What are approximates? Moisture, it's the weight loss after drying. There's a lot of literature and so forth about this. There are general procedures for doing that, pros and cons of that. Crude protein, nitrogen by 6.25 is generally in the U.S. what's used, but 6.25 is a general number. It's not specific for any particular crop per se, and it's debatable about this figure. We haven't had any discussion on that here, but there's other figures that are used. And those of you may recall with the episode with melamine, we had the problem because they're looking at nitrogen, not protein. So when you multiply the nitrogen 625, it gives you a crude protein, which means it's an estimate. which means it was a bad estimate because it was mostly non-protein nitrogen, which is not protein. Crude fats, usually ether extractable. Ash is remainder after combustion in a furnace at 500 degrees Celsius. So there's no organic matter left. Fiber, usually with approximate is crude fiber, but we'll say that's remainder after acid treatment and drying. But it's mainly a comparative figure now. If we look at crude fiber, is not meaningful except for literature comparison. All right. Even though you will find foods, they'll give a fiber, crude fiber, and you'll find feeds, they give crude fiber, but it seems that it got ingrained in the regulatory process and appears on the label and is not really meaningful nutritionally. Total dietary fiber is not digested by humans. So that in human nutrition is the important thing to look at, is total dietary fiber. Acid detergent fiber, which is cellulose plus lignin, and lignin are minus indigestible to animals. That's lignin is indigestible. Then you got, so acid detergent fiber is generally indigestible. Nutrient detergent fiber has the ADF fraction plus the hemicellulose that is digestible. It's important to animals. This is very important in animal nutrition and calculating diets, energy for the diets. What are the amino acids? Of course, they're the building blocks for making proteins. You can use a Lego set, put all those little pieces together, and you make a protein if you call each one of those an amino acid. And you link them all together, and you make a protein. So you have, in animal nutrition, you have important amino acids: arginine, histidine, isoleucine, lysine, leucine, methionine, plus cystine, phenalanine, threonine, valine, and tryptophan. In my elementary nutrition courses in college, you had A Hill MPB. How many remember that? There's a few people that do. So that's the arginine, histidine, isoleucine, and so forth. They're considered essential for non-ruminant animals. And of course, there's a difference between the ruminants and the non-ruminants. The non-ruminants are more like people that have simple stomachs, and the ruminants have 4 stomach compartments with a rumen, which is a big vat for fermenting the nutrient. Glysine, tyrosine, and serine are also important, particularly in poultry. Most limiting amino acids for animals are lysine, methionine, and threonine. That means in the diets of animals with the ordinary feed ingredients, those are the things most likely to be limiting. So it limits the full growth of the animal. What are fatty acids? Some fatty acids are essential in the diet for animals. Linoleic acid, linolenic acid. Others of interest are linolenic gamma, palmitic, and palmitoleic. All right? But if we're looking for swine diets, then we're worried about having enough of these 2. All right? So some of the other things can vary, the other fatty acids, and they really don't matter a whole lot in the diet. What are minerals? Major minerals include calcium and phosphorus, magnesium, potassium, and sodium. Trace minerals include iron, selenium, manganese, copper, and zinc. Other than calcium and phosphorus, the key minerals depend on what the feed from the unmodified plant is recognized for. So if you have one that's more recognized for iron or selenium. But there's a problem, like with selenium, the soil where the plant is grown highly influences the selenium content of the plant. And that's probably the case for a lot of the minerals. And in animal nutrition, these trace minerals are added as a routine based on the geographic location where the feed they're being fed and where the feed generally comes from. So it's quite common to add these trace minerals to the diet. In formulating a diet, phosphorus and calcium are always important. And from the standpoint of phosphorus, the anti-nutrient phytase is a very important part of the equation. Here in Maryland, we're in Virginia, but next state over in Maryland, there's a regulation that the swine and poultry farmers have to add phytase to the diet to release the phytin phosphorus that's tied up because of the problem of the phosphorus going into the waterways. Because the phytin phosphorus is not digested and is excreted by the animals and goes in the manure and then eventually winds up in the waterways and is causing a problem. So that's why, phosphorus is important, and with the equation of the phytase, phytin phosphorus. Other vitamins: fat soluble A, D, E, and K, water soluble B1, B2, B6, C, folacin, niacin, and pantothenic acid. And again, the key vitamins are dependent on the feed from the unmodified, what the unmodified plant is recognized for. But in animal nutrition, it's common to add these vitamins particularly with the ruminant, and with the non-ruminants, you're concerned about some of the water-soluble vitamins, and they're also added to the diet. So, I know in human nutrition we've talked about vitamin K. Alison Edwards could tell us more about that, but it becomes important in certain diets, and that with the soy diets, maybe that. Okay, so putting that aside, let's talk about generation of data. So we start with experimental design, as David told us. That was what we should start with, is your experimental design, to ensure that your study is designed to show what you think. So when you line up racers to a starting line, you like to have them both equal to start, so if there's a difference, that you can find that difference, and you don't have several other factors entering into the equation, confounding your results. We like the word compounding. Right? Okay. The GM plant variety and its parent. Now, the reason you select a parent, again, is you only want as few differences in the comparator as you can. So you'd like to have the only difference that you really want to look at is the new plant protein and the transformation event. So those are the only differences that you would hope would be in there, so you can say that would be what the effect comes from. In order to, and they do several field locations are selected representing the geographic area where the bioengineered plant is expected to be grown. So this gives you replication and gives you more confidence in the results. Within a location, the plots of lands are allocated to experiment. Usually 3 or more blocks containing 2 or more plots are formed to ensure that the treated and control plants are grown under the same condition. So it's a way of petitioning out the effects to ensure that you're only wanting to look at the intended effect here, or the effect of the new plant protein and the transformation event on the plant. So we're narrowing down the differences that could exist as much as we can. And you do that by blocking if you expect different parts of a field would have the soil would be different enough to cause a difference in the plant. So that's why you're putting them in blocks to narrow that differences down that might exist there. So maybe David told, did Fisher design? Did he design these block designs? Well, anyhow, it comes from good design. Okay, precautions are taken to ensure no cross-fertilization or other outside conditions influence the outcome. So, if you're growing corn, you don't want genes flowing in from close relative or the close proximity to influence how that corn is going to be fertilized and grown. So you eliminate all those that you can, so you're only looking at what you think you are. Harvest, storage, and processing and analyses. Harvest, in other words, you're going to harvest them in a way that you retain the integrity of the sample, all right? So you want to maintain integrity is very important. So it's got to be harvested so you maintain that. Composition analysis is conducted using validated analytical methods. So you got to be sampling to maintain the integrity, all right? You have to store it, you have to process it to maintain the integrity, and then you have to use the validated analytical methods to measure. So we make sure that we take everything out of it from the standpoint of variability that we can. Resulting data then is subjected to appropriate statistical analysis resulting in probability estimates. All right. Now, probability estimates that we talked about earlier are just estimates. They're no more than that. How they're interpreted is up to the evaluator. We'll talk a little bit more about that. Sponsors submit data and information on the safety and nutritional assessments of the new plant variety. This is called the biotech notification file. This is what the U.S. FDA process is. FDA evaluates all known hazards and for potential unintended effects on plant composition and nutritional properties. And this is aside from doing all the evaluation of the transformation event and insurance of the stability of the new gene in the plant, and to make sure that the methods used are recognized and are valid and everything else is done. So the submission that comes in can be quite thick because it has all that information in it. So it is a scientific document. It's not just a summary. Now, somebody might read the regulations thinking the summary just says, you know, it is just an abstract, but it's not. It has data in it, tables in it, it has raw data, has statistical analysis, has scientific tables, and what we say in the U.S. is that no hanging chads. Now, some of you may not know what a hanging chad is, but we go back to a presidential election several years ago, and there were, in Florida they had where they were punching cards, but they'd leave a little hole punch on the card, so it's called a Chad. All right, so and they were debating whether to count those votes or not. All right, so we're saying do not leave that Chad hanging. If you bring up a question, then you have to answer the question scientifically. So you just don't bring up a question and leave it hanging. You have to answer it in the submission. Okay, data presented. Data on each component will be assembled in a table form. As I said, the mean, average, P or F value, and literature ranges usually given. And we talked about the sources of databases yesterday. We had papers on that, what the source of the databases are. The open literature is also a source. And as well as we can find how valid the data in the literature is. To be consistent with recognized database, component values must be given on a percent of dry weight. And I asked a question yesterday. You cannot compare oranges and apples, all right? So if you really want to know what's in a plant, you get it down so it's on an equal basis. 9% protein in a 30% moisture is not the same as that in a 10% moisture, all right? So you got to get it on an equal basis if you want to compare it. You can, fatty acids are usually given on a percent of total fatty acids. Part of that's because the way that you separate out the oil, and then you run your chromatographic procedure on fatty acids, and that's generally what's acceptable to people in the literature. Do it on a percent of total fatty acids. And there are people that will make the case that's how the fat should be, and the fat total should come from that. But amino acids, they also given on a percent of total protein. That's given that we know how much protein is in the plant, and that goes back to that nitrogen times 625. We could have a whole seminar on what's the appropriate factor there. The expressed protein is not a critical feed considered. Expressed protein is not a toxin or related to a known toxin, or other components are not adversely affected. If there is no compounds produced expressed that would require food additive petition. In the U.S., say that we do not, the law doesn't provide for preclearance of food. And allows for preclearance of food additive. Difference between a food and a food additive. Food is something you eat, of course. Food additive is something you add to food. All right, that's the simplest term I can give because you go round and round in circles on some of that. But if it's can cause a safety problem and is not known to be safe, then we require a food additive petition. We've had one such petition with the NPT2 enzyme. But ordinarily, that's the Congress has given FDA the scientific authority to make this determination. And there's been court cases to prove that FDA has this authority. So that is the difference between the U.S. and a lot of regulators is that the U.S. has the scientific authority to make these decisions. Okay. The key nutrients, anti-nutrients, and natural toxicants normally present in the bioengineered plant are not adversely changed. So that's what we're talking about mainly in this talk. If we're looking at evaluate composition data, the first comparisons to the experimental comparator. Significant difference found. Compare a recognized database range. Recognized database include but not limited to OECD, USDA, ILSI, NARO, WHO, NRC, and peer-reviewed journals. If difference still exists, logical scientific justification must be presented involving safety data or new safety data must be generated. Weight of evidence. Conclusion are made, and Lynn talked about weight of evidence. Conclusion made on acceptability of a bioengineered plant variety based on all the submitted evidence. Valid scientific rationale is acceptable to bridge data. The compositional data, while there may be significant difference between the bioengineered plant and its comparator for some of the constituents, literature ranges can help guide whether further assessment is needed, or the plant composition is within expected range for the constituents. Results of FDA consultations for feed 1995 to 2012. I emphasize feed because we've had, okay. All right, so just to wrap up, we've done several consultations, 129 transformation events with all these different varieties. And the bottom line is no difference has been found.